Do we have to click the present button as well?
Yes,
Okay.
I will do that. Thank you.
Hello, everyone. Today, I'll be discussing my project, the identification of FDA-approved compounds that demonstrate new potential for reducing Ranidine receptor 1 leak. First, I'll start by describing my project summary. So my project question and hypotheses were as follows. The question was, which FDA-approved compounds show potential in mitigating Ranidine receptor 1 leak? And the hypothesis was multiple FDA-approved compounds will show potential in mitigating Ranidine receptor 1 leak. Now, the method I used in my project was a secondary data collation and analysis using FRET, or fluorescence resonance energy transfer, and functional assays, or Tritium labeled Ranidine assays. And the conclusion that I came to was that 13 compounds showed novel but varied potential in mitigating Ranidine receptor 1 leak. It's important to note that the potential was varied, as this will be important later in the presentation. So let's discuss some background information regarding the project. So the Ranidine receptor functions in muscle contraction and in muscle tissue. So we'll begin by going over the basics of muscle contraction. So first, an action potential travels down a motor neuron and reaches a muscle cell, in this case, a skeletal muscle cell. It causes an action potential in the skeletal muscle, which will then lead to calcium ions being released from a structure within the cell known as the sarcoplasmic reticulum. This calcium will be released and bind to muscle proteins, which will initiate the skeletal muscle contraction. On the right, you can see a diagram of this process. Now let's talk specifically about Ranidine receptor 1. Ranidine receptor 1, or Ranidine receptor isoform 1, is a calcium ion channel in skeletal muscle sarcoplasmic reticulum. So what it does is it releases calcium ion when it is appropriate, such as during muscle contraction. Regulators exist on this Ranidine receptor 1 protein as well. Some examples include FKBP and calmodulin. On the right is an image of a Ranidine receptor 1 protein that was imaged from rabbit skeletal muscles. So sometimes Ranidine receptor 1 can become dysfunctional and can leak calcium ions when it's not supposed to be doing so. This can be due to dysregulation by regulators that I previously described, such as FKBP and calmodulin. Diseases such as malignant hyperthermia and central core disease can result from these Ranidine receptor 1 leakages. These diseases are rare but very serious. Thus, it's crucial to identify drugs that will target the Ranidine receptor 1 channel in order to advance disease treatments and help patients have better outcomes. Here you can see an image of the Ranidine receptor 1 channel in its normal state, where it is not leaking calcium ions, and in its leaky state. Now, let's talk about some background information regarding a laboratory technique that's used in this project. Data from that technique is used in this project. FRET, or fluorescence resonance energy transfer, utilizes fluorescence to measure binding between two molecules. These molecules are tagged and then measured for their level of binding. In this case, Ranidine receptor 1 is the protein that's being gauged by this process. What's specifically being measured is the binding between the two regulators, in this case, FKBP and calmodulin. In general, higher FRET values imply tighter binding of regulators and thus a stronger regulation of the Ranidine receptor 1 channel. And thus, higher FRET values also imply a healthy Ranidine receptor 1 channel. Now let's talk about the second set of data that's used in this project. Tritium labeled ranidine assay Tritium labeled ranidine assays. Ranidine is a molecule that has a strong affinity for binding to the open channel state of ranidine receptor 1. Tritium is a common molecular tracer that's used in biology in order to detect where molecules can be. The level of tritium labeled ranidine that binds to sarcoplasmic reticulum directly represents the activity of that ranidine receptor 1 channel. Thus, high assay performance is directly correlated with high leakage or high channel activity. This is a strong predictor of ranidine receptor 1 activity and leak, and it is stronger than FRET. FRET also has a direct negative correlation with the tritium labeled ranidine assays. So an increase in FRET values generally correlate with a decrease in channel activity. Let's discuss the method used in this study now. First, calculations were completed on compounds data to identify the compounds that statistically significantly decreased channel activity in tritium labeled ranidine assays at unhealthy conditions and at low calcium ion levels. Unhealthy conditions signify a disease state of the body, and low calcium ion levels signify a non-contractile period of a non-contractile event. Then FRET datasets were correlated with tritium labeled ranidine assay data to determine the FRET dataset that was the best predictor of decreases in the tritium labeled ranidine assay activity. And it was found to be biosensor 1 at 120 minutes of incubation. Next, compounds were identified that were predicted by the best FRET dataset, which was biosensor 1 at 120 minutes incubation, but were not tested in the assay. Here is some data from the ranidine assays. As you can see, they were tested at unhealthy conditions at low calcium ion levels. For example, you can see some compounds that had a statistically significant decrease in ranidine receptor 1 channel activity, such as anastrozole, Reprodil, retigabine, lornoxicam, and ceforanide, as well as dantrolene, which was used as a control. It is important to note that some compounds had a statistically significant decrease as well as a statistically significant increase at different concentrations of the compounds added. This is important later in the presentation. Now, here's some data that the best predictor, or biosensor 1 at 120 minutes incubation time, is used. So as you can see, the biosensor 1 at 120 minutes predicted correctly every single instance of statistically significant decreases in the assay activity. And it had the lowest error rate of all of the biosensors tested. In addition, some biosensor 120 data points did not become tested in the assays. Thus, those show potential to decrease channel activity, although they weren't tested yet. They're highlighted in blue here. Now, let's summarize the results. Anastrozole, Reprodil, hydrochloride, retigabine, lornoxicam, dantrolene, and ceforanide statistically significantly decreased the ranidine receptor 1 channel activity in tritium labeled ranidine assays at low calcium ion levels and in unhealthy conditions. Furthermore, nintedanib, palmitine, palmitine chloride, calcipotriene, diacerine, quercetin, harmaline, and dihydroxylene sulfate, and procarbazine hydrochloride were not tested in the assay, but did increase FRET values for biosensor 1 at 120 minutes. Now I'll discuss the conclusions of these results. So as we can see here, there were 13 FDA-approved compounds organized by potential of decreasing leak in ranidine receptor 1. In the first list are the compounds that in the assays only statistically significantly decreased the channel activity. In the second list are compounds that in the assays had a statistically significant decrease in the channel activity and a statistically significant increase as well. And third, we have the compounds which showed potential in reducing ranidine receptor 1 activity, but were not tested. The reason for this ranking is that in the first list, there were no statistically significant decreases. What this means, no statistically significant increases. What this means is that the drug only caused a down regulation of the ranidine receptor. Thus, it shows the most potential to have an effect in the human body. The second list shows a mixed bag of results. These may show potential, but more research is needed. And finally, in the third list, these drugs were only tested in FRET, but they increased the regulation, which in clinical science is known to decrease the channel activity, but we still need to test these compounds in order to be sure. Let's talk about some of the implications of this study. So drugs identified previously may serve as treatments in diseases such as central core disease and malignant hyperthermia due to their potential to reduce the ranidine receptor 1 activity with further testing, such as in situ tests of muscle fibers and animal studies. Some of the practical implications include pharmaceutical companies, which may rebrand some of these drugs after further testing to treat diseases such as central core disease and malignant hyperthermia. Some of the research implications include biosensor 1 being screened at 120 minutes was a very, very accurate gauge of assay activity. So in the future, it can be used to propose drugs from the FRET screen to the assay tests. The hypothesis that multiple FDA-approved compounds will show potential in mitigating ranidine receptor 1 leak was confirmed. Thank you.
Pramit. So as you know, there are oral defense questions, and you've had an opportunity to prepare for those. I will be asking you one from each category: the research process, your depth of understanding, and reflection on the inquiry process. My first question for you, you have alluded to this, but I'm wondering if you could tell me a little bit more about how you determined which results generated by your research method were most important in forming your new understanding.
So some of the results were not statistically significant, such as some results didn't statistically significantly decrease the assay activity, or some of the results didn't statistically change the FRET data. So sometimes data needed to be filtered, and that's how the conclusion was reached.
My next question, can you tell me a little bit more about how your findings provide direction for future research in the field?
Sure. So some of the methods that I used in my research revealed some important results. So as I mentioned earlier, biosensor 1 at 120 minutes was a very good gauge at predicting how the drug will perform in the functional assays. So now, if a researcher has a massive amount of drugs and they need to test these drugs and propose certain ones for assay testing, we can use this analysis of the FRET data to propose further research.
sense. Thank you. And then my last question for you. What was the most important research skill that you developed as a result of this process, and how might you apply it to future endeavors?
Sure. So the most important research skill that I gained from this was asking questions and seeking help when I needed it. So whenever I had a question about some data that I had, I would go to Dr. Rebek, who is my mentor, and I would ask her questions and she'd explain those, and that would cause me to gain a greater understanding of my research project.
Excellent. Well, Pramit, that finalizes the formal part of your presentation with oral defense today. Congratulations.